NAD plus supplementation rejuvenates aged gut adult stem cells. Masaki Igarashi abstract. The tissue decline due to aging is associated with the deterioration of adult stem cell function. Here we show the number and proliferative activity of intestinal stem cells, ISCs, but not Paneth cells decline during aging, as does ISC function assessed ex vivo. Levels of SIRT1 and activity of mTORC1 also decline with aging. The treatment with the NAD plus precursor nicotinamide riboside, NR, rejuvenates ISCs from aged mice and reverses an impaired ability to repair gut damage. The effect of NR is blocked by the mTORC1 inhibitor rapamycin or the SIRT1 inhibitor X527. These findings demonstrate that small molecules affecting the NAD/SIRT1/mTORC1 axis may guide a translational path for maintenance of the intestine during aging. 1. Introduction. Aging is one of major risk factors of adult-onset disease such as cancer, diabetes, Alzheimer's and Parkinson's disease, and cardiovascular disease, Nicolai and Partridge. Adult tissue homeostasis is controlled by adult stem cells, which are continuously proliferative and maintain the tissue, hematopoietic and intestinal stem cells, ISCs, or quiescent and induced by tissue injury, muscle, liver, and neural stem cells. Chandel, Jasper, Ho, and Paseghi. Previous studies have described the aging-related deterioration of adult stem cell function. In certain cases, this decline could be attributed to a loss in the activity of one of the sirtuins, which are nicotinamide adenine dinucleotide, NAD+, dependent decylases that regulate aging and age-related diseases, GURANT. For example, in hematopoietic stem cells, a reduction in SIRT3 or SIRT7 activity compromises the regenerative capacity of HSCs in aging mice, Brown et al., Morin et al. In muscle stem cells, MUSCs, reduced amounts of NAD+ and the associated decline in activity of SIRT1 are determinants of aging-related decline. Notably, treatment with NAD+ precursor nicotinamide riboside, NR, Canto et al., induces the rejuvenation of MUSCs in aged mice and extends the lifespan of the animals, Zhang et al. The rapid turnover of the intestinal epithelium is sustained by ISCs. Previous studies of aging of ISCs mainly come from studies on the intestinal epithelium of Drosophila, Bitto, Hochmuth, and Jasper. During aging, the number and activity of cells that express stem cells marker in Drosophila midgut increase due to an environmental challenge or tissue injury, Bitto, Hochmuth, and Jasper, Choi, Kim, Yang, Kim, and Yu, Hochmuth et al. In mammals, LGR5 expressing cells in the base of the crypt constitute the majority of ISCs under normal conditions, Barker, Tan, and Clevers, Barker et al. Recently, it was reported that aging results in a decline in ISCs function in mammals and WNT signaling ameliorated the imperative function of aged ISCs, Mihailovan et al., Nolaparty et al. Calorie restriction, CR, has been shown to trigger the release of cyclic ADP-ribose from the ISC niche, the Paneth cells, Yilmaz et al., to drive a pathway of signaling in LGR5 expressing ISCs to promote their expansion in cell number. This pathway involves SIRT1 activation due to increases in the NAD+ biosynthetic enzyme, nicotinamide phosphoribosyltransferase, NAMPT, SIRT1 decetylation of S6 kinase 1, S6K1, and consequent phosphorylation of decetylated S6K1 by mTORC1. Igarashi and Gurant. Notably, mTORC1 activation by CR and ISCs is opposite to its observed repression during CR in many differentiated cells, including Paneth cells. As predicted by the model, the mTORC1 inhibitor rapamycin and genetic ablation of SIRT1 suppress the effect of CR on ISC expansion, Igarashi and Gurant. Here we show the murine ISC number in vivo and functional activity ex vivo decline due to aging. In striking contrast, the number of Paneth cells in mice and their function in supporting ISCs are not impaired by aging. NAD+ supplementation by the NAD+ precursor NR can rescue these ISC defects and restore a youthful number and function of ISCs. Thus our findings suggest a translational path of NAD+ replenishment for rejuvenating the aged gut. 2 results. 2.1 The ISC pool decreases due to aging. To assess the effect of aging on gut homeostasis, we compared the histology of small intestine in young, 3-5 months old, and old, more than 24 months old, C57BL/6 mice. Morphologically, aging induced an increase in villus length and crypts showed a trend to a smaller size, which did not reach significance, Figure 1a. In agreement with the increase in villus size, aging increased the numbers of differentiated cells of the gut, absorptive enterocytes, goblet cells, and chromogranin A plus enteroendocrine cells, supporting information Figure S1ac. Figure 1 aging reduces ISC number in vivo. H&E staining images and the quantification of the crypt size and villus size in the intestine of young, 3 months old, and old, more than 24 months old, C57BL/6 mice, 6 mice per group. Approximately 50 crypt villus units per mouse. All histological images in this and subsequent figures are typical of numerous tissue samples analyzed. BRDO staining images and the quantification of BRDA positive crypt-based columnar, CBC, cells, arrowheads, adjacent to Paneth cells at the bottom of crypts is assessed 2 hours after the injection of BRDA, 3 mice per group, 50 intact well-orientated crypts per mouse, C. In situ hybridization images of ALFM4 mRNA and the quantification of ALFM4 plus ISCs. Arrowheads, 3 mice per group. Approximately 25 intact well-orientated crypts per mouse, D. Lysozyme staining image, red, lysozyme, blue, DAPI, and the quantification of lysozyme-positive Paneth cells, 3 mice per group. 50 intact well-orientated crypts per mouse. Original magnifications, X100, X400, B, and X200, C and D. Scale bar, 100 microm, 25 microm, B, and 50 microm, C and D. Values represent the mean plus or minus MP, 0.05, P, 0.01, t-test. ISC, intestinal stem cell next, to test whether aging influences the population of proliferative cells in young versus old mice. 2 hours BRD labeling was performed in crypts, which marks ISCs and their immediate descendants, transit amplifying, TA. Cells, Figure 1b and supporting information Figure S1d. There was no difference between young and old mice in the incorporation of BRD into total crypt cells, ISCs and TA cells, supporting information Figure S1d. However, there was a decreased BRD when incorporation into the crypt-based columnar, CBC, cells, which are stem cells wedged between Paneth cells, Figure 1b, Barker, Van Oudenerden, and Clevers, indicating that proliferation of ISCs decreased in old mice compared to young mice. Next, to investigate the number of ISCs in young versus old mice, we performed in situ hybridization, ISH, for olfactomidin 4, ALFM4, which is a marker of ISCs, Van der Flyer, Hegebarth, Stange, Van de Wettering, and Clevers, Flyer et al. The number of ALFM4 plus positive ISCs in the crypts of old mice decreased compared with young mice from about 5 cells per crypt to about 3 cells per crypt, Figure 1c. A similar finding was noted using FACS analysis of mice expressing LGR5-GFP plus cells to count ISCs, supporting information Figure S1e. Paneth cells are niche cells which are adjacent to and support the proliferation of ISCs, Sato et al. Particularly, ISC proliferation in CR depends on the cyclic ADP ribose secreted from Paneth cells, Igarashi and Guerrant, Yilmaz et al. However, we did not find any difference in the number of Paneth cells, lysozyme positive, between young versus and old mice, Figure 1d. These results all indicate that the number of ISCs in old mice decreases and the villi length increases, suggesting that self-renewal of ISCs is reduced and differentiation of ISCs is consequently increased in old mice. 2.2 Aging degrades the formation of intestinal organoids from ISCs. To further investigate the effects of aging on the proliferation of ISCs, we isolated crypts from young and old mice, dispersed the cells, and seeded them on mat-rigel to obtain organoid colonies, Igarashi and Guerrant, Sato and Clevers. Each organoid colony is seeded by a single ISC and gives rise to all the differentiated cells of the gut in the colony. This assay thus enumerates the functional ISCs ex vivo. Crypts from old mice formed fewer organoid colonies and showed a decreased number of buds, which is another indicator of stem cell function, compared to those from young mice, consistent with the in vivo data. Figure 2 and supporting information Figure S4. Next, to assess organoid-forming ISCs more accurately and address how the ISCs and Paneth cells interact functionally, we isolated LGR5 positive ISCs and Paneth cells each to 95% purity from young and old LGR5 Eggpires Creer2 mice. As described previously, Igarashi and Guerrant. A total of 2,000 cells were plated of each cell type and in the standard media containing glycogen synthase kinase
3 Vita,
GSK3 Greek letter beta, inhibitor CHIR 99021, CHIR, which is known to induce
Vita,
catenin and thus stimulate organoid formation. Igarashi and Guerrant, Yanet al. LGR5 positive ISCs isolated from old mice formed fewer and smaller colonies on day 5 of culture and fewer organoid colonies with a decreased number of buds on day 9, compared with those from young mice. Figure 2b and supporting information Figure S2 and S5b, C. By combining young or old ISCs and young or old Paneth cells, we determined that the Paneth cells from old mice were as functional as young Paneth cells in stimulating organoid colony formation in co-culture with young ISCs and with or without CHIR. Figures 2c, supporting information Figures S2b and S5d. Old ISCs were similarly defective in organoid formation with either young or old Paneth cells. Thus, this functional ex vivo assay for ISC function and niche stem cell interaction demonstrates a defect in old ISCs but not old Paneth cells. Figure 2 aging reduces the formation of intestinal organoids from ISCs ex vivo. Crypts from young and old mice were cultured in mat-rigel to allow ISCs to form organoid colonies. The number of colonies was assessed at day 5, 7-8 Wells group, the sum of 2 different experiments. The picture shows colonies cultured from young crypts and old crypts. B. ISCs were isolated from young and old LGR5 Eggpires Creer2 mice, 95% pure. And 2x103 cells were cultured in the absence of Paneth cells in culture medium containing 10 micron CHIR. The number of colonies was assessed at day 5, 11-15 Wells group, the sum of 3 different experiments. The picture shows colonies from young ISCs and old ISCs culture in the absence of Paneth cells. C. ISCs and Paneth cells were isolated from young and old LGR5 Eggpires Creer2 mice. And 2x103 cells each were co-cultured in the medium containing 10 micron CHIR. The number of colonies was assessed at day 5, 3-4 Wells group. Original magnifications, X100, and B. Scale bar, 100 micron, and B. Values represent the mean plus or minus MP, 0.05, P, 0.01, P, 0.001, T-test. ISC, intestinal stem cell to gain insights into the mechanisms driving loss of organoid formation in old ISCs. We focused our analysis on gene networks previously associated with ISC stemness, Igarashi and Guerrant, and found a significant reduction in SIRT1 protein in old crypts and ISCs, supporting information Figure S2c, D. Also, we observed a trend to a reduction in S6 phosphorylation in old crypts and old ISCs, indicative of a lowering in SIRT1/mTORC1 activity, Figure 4c, D. Notably, our NSEC analysis of young and old ISCs did not reveal significant differences in gene expression, suggesting that relevant changes occur post-transcriptionally, supporting information Figure S3. 2.3 Nicotinamide riboside restores the colony formation in aged mice. In order to probe the role of the SIRT1/mTORC1 axis ISC aging, we tested whether NAD plus supplementation affects the function of old ISCs ex vivo. Thus, we treated crypts from young and old mice with NR, a NAD plus precursor, which increases intracellular NAD plus levels and activates SIRT1, Canto et al. NR treatment completely rescued the diminishment in colony formation efficiency and number per organoid of differentiated buds in old crypt-derived organoids, while having no significant effect in the colonies from young crypts. Figure 3 and supporting information Figure S4a. To address in more functional detail the role of the SIRT1/mTORC1 pathway in the N-rejuvenated ISCs, old crypts were incubated with NR plus the mTORC1 inhibitor rapamycin or the specific SIRT1 inhibitor X527. Rapamycin or X527 treatment of old crypts blocked the rescue of organoid formation by NR treatment, Figure 3b, c, while having a minimal effect on N-retreated young crypts, supporting information Figure S4b, c. Next, to be certain these effects occurred specifically in the ISCs, we purified LGR5 plus stem cells and carried out parallel experiments to the studies using crypts. We found with the pure ISCs that NR again increased organoid colony formation by old ISCs, and that this increase was blocked by rapamycin or X527 treatment, Figure 3d, e, while having a minimal effect on N-retreated young ISCs. Supporting information Figure S4d, e. These studies show that ISC functional decline with aging can be rescued by NAD plus replenishment via NR and that the effect is intermediated by SIRT1/mTORC1 signaling pathway. Figure 3 Nicotinamide riboside, NR, restores the colony formation in aged mice. Isolated young or old crypts were cultured in medium with or without 1 mm NR as indicated, 5-6 Wells group. Representative images of the formed organoids at day 5 and the quantification of organoids number. Original magnifications, x50. Scale bar. 100 micron, b. Isolated old crypts were cultured in medium with or without 1 mm NR and 1 mm rapamycin as indicated, 5 Wells group, c. Isolated old crypts were cultured in medium with or without 1 mm NR and 1 micron X527 as indicated, 4-5 Wells group, d. Isolated old intestinal stem cells, ISCs, were cultured in medium with or without 1 mm NR and 1 mm rapamycin as indicated, 4 Wells group, e. Isolated old ISCs were cultured in medium with or without 1 mm NR and 1 micron X527 as indicated, 3 Wells group. Values represent the mean plus or minus MP, 0.05, p. 0.01, p. 0.001, t-test 2.4 NR treatment restores ISC number in aged mice in vivo. We wished to test whether the decline in ISC number in old mice could be reversed by NAD plus replenishment. Thus, we tested whether NR treatment affects the number of ISCs in vivo in young or old mice with a 6 weeks treatment of drinking water containing NR. 500 mg/kg body weight, or vehicle, Figure 4. First, we tested whether NAD plus supplementation affects the function of ISCs derived from old mice, but not Paneth cells in vivo. In vivo, NR treatment completely rescued the diminishment in colony formation efficiency in old crypts and old ISCs, supporting information Figure S5ac. On the other hand, by combining ISCs and Paneth cells purified from young or old mice, we determined that NR treatment affects ISCs rather than Paneth cells, supporting information Figure S5d. Next, we observed that the elongation of villi induced during aging was abrogated by NR treatment, supporting information Figure S6a. To investigate how NR treatment influenced the activity and number of ISCs in young and old mice, BRD labeling and ISH for the ISC marker ALPHM4 were performed. NR treatment completely rescued the decrease of BRD uptake into CBC cells, Figure 4a, and, more importantly, the decrease of ALPHM4 plus positive cells, Figure 4b. Again, NR treatment had minimal effects on the expansion of ISCs in young mice, Figure 4a, b. Moreover, we found a significant increase in SIRT1 protein and S6 phosphorylation in N-retreated old crypts and old ISCs, confirming that ISC functional improvement by in vivo NAD plus replenishment activates the SIRT1/mTORC1 signaling pathway, Figure 4c. DN supporting information Figure S6b. Figure 4 NR treatment restores ISC number in aged mice in vivo. BRDA staining images and the quantification of BRDA-positive CBC cells, arrowheads, in young or old mice administered with a vehicle or NR, 500 mg/kg. In drinking water for 6 weeks, mice is assessed 2 hours after the injection of BRDA, 3-5 mice per group, 50 intact well-orientated crypts per mouse. B. In situ hybridization images of ALFM4 and RNA and the quantification of ALFM4 plus ISCs, arrowheads. In young or old mice administered with a vehicle or NR, 500 mg/kg, in drinking water for 6 weeks, 8 mice per group, 50 intact well-orientated crypts per mouse. C. In situ phospho-S6, PS6, staining images, green, PS6, blue, DAPI. In the intestine of young or old mice administered with a vehicle or NR, 500 mg/kg, in drinking water for 6 weeks and the quantification of relative signal intensities per area in crypt, 5 mice per group. At least 4 different image fields per each sample were quantified by image JY, young, O, old. Original magnifications, X400, X200, B, and X100, C. Scale bar, 25 micron, 50 micron, B, and 100 micron, C, D. Immunoblotting of PS6 or S6 and ISCs isolated from young or old LGR5 EGFP IREs CREAR 2 mice administered with a vehicle or number. Values represent the mean plus or minus MP, 0.05, P, 0.01, P, 0.001, T-test, CBC, crypt-based columnar, ISCs, intestinal stem cells, NR. Nicotinamide riboside 2.5 NR rescues functional defects in aging mouse gut. Finally, we wished to determine whether the reduction in ISC number would correspond to functional defects in the aging gut. The colon is sensitive to dextrin sulfate sodium, DSS, which causes injury requiring tissue regeneration by colonic crypts, Yui et al., Zhao et al. Therefore, DSS colitis is a good model for investigating the regenerative function of ISCs. First, to investigate the effects of aging on the proliferation of colonic stem cells, we isolated crypts from colon of young or old mice and seeded them on mat-regel to obtain sphere colonies, O'Rourke, Eckermann, Dow, and Lowe. Colonic crypts from old mice formed as many but smaller colonies compared with young colonic crypts on day 5, figure 5. Furthermore, colony formation efficiency from old colonic crypt significantly decreased after an additional 5 days of secondary passage, figure 5b. NR treatment completely rescued those defects in colonies formed from old colonic crypts, while having a minimal effect in the colony formation from young colonic crypts, figure 5a, b. Figure 5 NR treatment reverses functional decline in ISCs during aging in vivo. Isolated young or old colonic crypts were cultured in medium with or without 1 mm NR as indicated. The diameter of more than 70 colonies per each group was analyzed on day 5. B. The number of colonies cultured in medium with or without 1 mm NR was assessed after 6 days of primary culture and an additional 5 days of secondary passage. 4-5 wells per group. Original magnifications, X50, and B. Scale bar, 100 micron, and B, C. Young and old mice administered with a vehicle or NR, 500 mg/kg, in drinking water for 6 weeks were treated by 1.5% DSS for 5 days followed by 3-day water feeding before being sacrificed, 5 mice per group. The histaining of distal colon, 10 mm from middle, tissues and the percentage of damaged colonic crypts are shown. Bottom, the enlargement of the squared area. At least 3 different X40 image fields per each sample were quantified. Original magnifications, X50 and X200, enlargement. Scale bar, 250 micron, enlargement. Values represent the mean plus or minus MP, 0.001, T-test. DSS, dextrin sulfate sodium, ISCs, intestinal stem cells, NR. Nicotinamide riboside to test whether old mice were more sensitive to DSS. Mice were administered 1.5% DSS for 5 days followed by 3-day recovery periods before analysis. Old mice were found to be much more susceptible to disease-induced damage than young mice, Figure 5c. To test the ability of NR to rescue this deficit, young and old mice pre-treated by NR vehicle for 6 weeks before DSS challenge. Indeed, NR treatment suppressed the increase in susceptibility to DSS to levels observed in young mice, Figure 5c. The above data demonstrate that NR rescues damage repair defects in the aging gut, likely by restoring the ISC pool to numbers found in young mice. 3. Discussion. The deterioration of organ integrity with aging can be in part attributed to a decline in adult stem cell number and function. Here we investigate the fate of ISCs in aging mice, both in terms of cell number and cell function. Our findings clearly show a decline in ISC cell number in 24 months or older mice. In contrast to ISCs, niche cells or Paneth cells showed no decline in cell number in old mice. One recent paper did not find a decline in ISC number in old mice, Nolaparity et al., but another study, Mihailov et al., did. This disparity may reflect different strains, housing conditions, or methods. Similarly, we assessed the abilities of ISCs and Paneth cells to function ex vivo by their ability to give rise to organoids in a mat matrix. The organoid assay again revealed a functional defect in ISCs but not in Paneth cells from old mice. Other studies have shown the benefits of NAD plus replenishment by the NAD plus precursors NR and NMN in metabolic function, Kanto et al., Yoshino, Mills, Yoon, NMA, and stem cell number, Zhang et al., especially musks. In addition, SIRT3 upregulation has been shown to rescue aging deficits in hematopoietic stem cells, Brown et al. The fact that we observed rescue of ISC number and function by NR expands our understanding of stem cell aging. Most interestingly, rapamycin blocked the rejuvenation of ISCs by number. This is because SIRT1 and mTORC1 are both activated and function sequentially in response to the Paneth cell signal Cadber, Igarashi and Guerrant. Indeed, SIRT1 desetylates S6K1 fostering its phosphorylation by mTORC1 and the activation of translation accompanied by an increase in ISC number, Igarashi and Guerrant. Our findings suggest that NAD plus replenishment may be a viable strategy for maintaining gut health with aging. Nolaparity et al. And Mihailov et al. Also reported a decline in organoid formation in crypts derived from old mice, and this could be rescued by elevating WTN signaling, Nolaparity et al., or activating a fatty acid oxidation program. Mihailov et al. It is thus likely that multiple signaling pathways affecting ISCs can be triggered to rejuvenate the aging gut. It is noteworthy that mitochondrial defects are a common thread in the decline of adult stem cells. Similarly, defects in DNA repair leading to premature aging also triggers NAD plus depletion via PARP and leads to defective mitochondria, Fang et al. In both aging stem cells and cells with DNA repair deficits, the mitochondrial defects can be rescued by NAD plus replenishment and SIRT1 activation. Along with our findings in the gut, these results all suggest that NAD plus replenishment may be a viable strategy to slow aging in a variety of tissues. Our findings also raise the possibility that anti-aging interventions, like rapamycin, may have beneficial consequences in some contexts but actually be deleterious in others. It will be interesting to see whether small molecule interventions, for example, rapamycin, metformin, NR, show different tissue selectivity in their abilities to improve function during aging. It is possible that different individuals will respond best to one or another intervention, depending on which organ is most vulnerable to aging in that person. Human trials over the next few years may shed light on this important question. For experimental procedures. 4.